All right, welcome everyone. It's great to see you on this very special day. It's a privilege to be in this position. I'm Rich Lenski. I'm a professor in microbiology here at MSU, and I've had the privilege of working with Nkrumah for the last 6 years. So let me begin with a warm welcome to all of you, especially to Nkrumah's family and friends. And the theme of today, I'm going to say, is about growth. It's about growth of bacterial cells and populations, the growth of scientific knowledge that comes from studying bacterial growth, and especially the growth of an individual scholar, Nkrumah Grant, who has produced this new knowledge through his skill, creativity, and hard work. A dissertation defense is the high point of academic life. The PhD is the highest degree that a university bestows. It takes years of advanced coursework and, more importantly, the production of an original body of knowledge. It's not only learning what is already known, but creating the new knowledge that will be read by future students and built upon by future scientists. So here's a little background about Nkrumah and his accomplishments. He received his Bachelor of Science degree from Grand Valley State University in 2014. Here at MSU, he's taken enough courses to satisfy not one, but 2 sets of requirements. So today he is a candidate for a dual PhD in the Department of Microbiology and Molecular Genetics and in the graduate program in Ecology, Evolutionary Biology, and Behavior. And Nkrumah has garnered a number of awards, of which I'm just going to mention a few. He was a McNair Scholar at Grand Valley. He was recruited to MSU with an Academic Achievement Fellowship. He received honorable mention for a National Science Foundation Fellowship. And this past spring, Nkrumah received the Ralph Evans Award from the Microbiology Department for his contributions to understanding microbial evolution and the ecological and genetic processes that produce evolutionary change. One of the currencies of science is presenting research at conferences, and Nkrumah has done many of those. Another is the publication of research findings in scientific journals. Nkrumah was a co-author on one paper a few years ago, and his dissertation chapters that you'll hear about today form the basis for 3 more publications, one of which is currently in review. Another is going to be submitted next week. That one came out of a talk Nkrumah gave, and he was invited to submit. And a third will be wrapped up and submitted soon after that. And on top of his dissertation papers, he's working on at least 2 others dissertation, but which already build on his dissertation work. And I saw that he posted one of those, I think, just this morning or yesterday. Throughout all of this, Nkrumah has been a generous and active member of my lab group, of the Beacon Center, of the Microbiology Department, of the University as a whole, and the field of microbiology. This again, the next program he serves on the College of Natural Sciences Diversity, Equity, and Inclusion. His outstanding career as a postdoctoral research scientist in the lab of Professor Chris Marks at the University of Idaho, and he'll start doing that this fall. So without further ado, here's Nkrumah to tell you about his dissertation research, which is entitled Deconstructing the Correlated Nature of Ancient and Emergent Traits, an Evolutionary Investigation of Metabolism, Morphology, and Mortality. So take it away, Nkrumah. And unmute yourself.
Great. Can everybody hear me well? I kind of lost you all for a brief moment. We good?
We're good.
All right, great. All right, so thank you, Rich, for the introduction, and thank each and every one of you here today joining me on this momentous occasion. I have a lot of ground to cover today, so I'm just going to get started with my dissertation talk entitled Deconstructing the Correlated Nature of Ancient and Emergent Traits, an Evolutionary Investigation of Metabolism, Morphology, and Mortality. So blue-eyed cats with white fur are adorably cute. However, just because this cat has these traits, it is probably also invariably deaf. Hairless dogs, they are hideously amusing to me. And even more so when you ask them to open their teeth, the fact that they're jagged. These are examples of correlations that Darwin and his contemporaries noted in the 1800s leading Charles Darwin to note in his opening chapter of The Origin of Species that if man goes on selecting and thus augmenting any peculiarity, he will almost certainly unconsciously modify other parts of the structure owing to the mysterious laws of the correlation of growth. See, the concept of the gene was not at all known during Darwin's time. Gregor Mendel's work, for example, was not published until 1866. Yet there was still this understanding that something within the organism was contributing to the observed phenotype. Now, to fast forward to the 21st century, we now understand that underlying these phenotypes are genes and the interactions between them. Take here, for example, this gene regulatory network for E. coli, where each blue node represents a gene, and each red node represents a transcription factor acting on that gene. And what I want you to appreciate about this is that it's this network that has complicated efforts to understand how the genotype maps with phenotype. Now, if we can understand how the genotype maps with phenotype, perhaps we can predict what phenotypes are likely to respond to change in selection pressures, including climate change. And then perhaps we can also use the information gathered from the natural genetic architecture as design principles to generate genetically and environmentally robust organisms. Now, one cannot go back in time and witness how these correlations came about. However, one can examine how these correlations change with time and in response to changing selection pressures or to different perturbations, given that the individual knows a certain genotype and can track changes to that genotype and associated phenotype with time. And it's the framework of experimental evolution that makes this possible. So for those of you who are unfamiliar with this, experimental evolution is the act of imposing selection pressures on an organism in a controlled laboratory environment. And these experiments are oftentimes done with single-cell organisms such as yeast and prokaryotes, but can also be done computationally owing to the fact that these model organisms have rapid generation times, simple genomes, and due to their amenability to preserving intermediate phenotypes. Now, one such long-term evolution experiment was established in 1988 by Richard Lenski with 12 populations of E. coli, all started from a single common ancestral strain. There are no prophages in the system, no horizontal gene transfer, and no sex. So evolution proceeds purely by the forces of mutation, natural selection acting on those mutations, and genetic drift. Now, the experiment in itself is simple in design. Every 24 hours, 1% of culture, exhausted of nutrients, is transferred into fresh growth medium. The bacteria undergo 6.6 generations per day, such that by 70,000 generations, such that by the year 2020, the bacteria have been evolving for greater than 70,000 generations. Now, whole population samples and clones from each population have been frozen every 500 generations. This creates a frozen fossil record from which evolution can be studied retrospectively in adaptive and non-adaptive environments using metagenomic sequencing, as well as using evolutionary neutral marker that was present in the ancestor that allows us to discern between the ancestor and the evolving populations just by virtue of mixing them together and counting colonies on plates. Now, today I'm going to tell you how I use this sweet old LTEE to study M&Ms. And of course, this is an acronym. I don't think EHS would have loved to see me with M&Ms in the lab. So in the chapter 1, I'm going to tell you about metabolism, more specifically about the genomic and phenotypic consequences of 50,000 generations of relaxed selection on anaerobic growth. In chapter 2, I'll talk to you about cell size and shape evolution and their relationship with fitness. And then lastly, I'll open the curtain in chapter 3 and talk to you about mortality and the concomitant evolution of a metabolic innovation in cell death. So with that being said, shall we turn the page on chapter 1, metabolism. So oftentimes as evolutionary biologists, we like to think about things in terms of selection. Positive selection is something that comes to mind. But one unappreciated mode of selection or lack thereof is relaxed selection, which can be defined as the weakening or complete removal of selection on a trait. Now, relaxed selection in itself predicts trait loss. Take here, for example, this species Asianus mexicanus, which is derived into a surface dwelling form and a cave dwelling form, which lacks pigment in its skin and also lacks eyes altogether. Now, from an evolutionary biology perspective, this is what we term here as consider a trade-off. And trade-offs occur primarily by two mechanisms: antagonistic pleiotropy, whereby mutations or traits under selection in one environment come at the expense of other traits, or mutation accumulation, whereby genes that are not being used accumulate mutations, and then those genes, they break with time. Now, the effects of relaxed selection are only observed when the selection pressures are reintroduced. So obviously, in a cave where one doesn't need to be seen or doesn't need to see its food because it's dark, it may be beneficial to lose those traits. However, if you put this strain in an environment that it needs to evade predators, for example, obviously it's not going to do well. It can't see them and it's easily visible. So relaxed selection generally can be described by saying you don't use it, you lose it, right? Well, I challenge this question with, or do you? And to go back to that network I showed you earlier, my thoughts are traits with more genetic interactions safe, right? Good. So relaxed selection is a dominant feature of the LTEE in itself. E. coli, they don't live in flasks. They live between the hosts and environments, and they constantly cycle with time. And in one way is the fact that E. coli is a facultative anaerobe, which means that it could grow in the presence or absence of oxygen. But by virtue of the constant aeration that occurs by the flask constantly shaken in the incubator, metabolic plasticity, or the ability to switch between aerobic and anaerobic metabolism, has not been occurring. So the motivating question for my chapter 1 was, is metabolic plasticity retained? And I want to know if I can understand this from a genomic and phenotypic basis of the trait. So in order to do that, I went back into the literature, and we found that took advantage of publicly available genome sequences for the strains. So in 2017, Ben Good at Stanford, he deep sequenced all of the 12 populations through about 60,000 generations. And obviously, E. coli has been a model of physiology since its discovery by Theodore Escherich in 1885. So there's plenty of data on it. And I went back into the literature, and I was able to find genes from E. coli that were expressed solely in an anaerobic environment or solely in an aerobic environment. And what I can do with these genes, I could then essentially ask the metagenomic data sequences, what do the mutations look like in those genes? So that's a simple thing where I can use information about the sequencing in our lab and use information about the transcriptional profile of E. coli grown in anaerobic or aerobic environments, and I can marry them together and ask these questions. And just to set the stage a little bit further, I'm going to blow up this picture. So here I'm showing you each one of the 12 populations in each one of the panels. Each line represents a mutation with the time that mutation that was detected indicated by a star. And what I hope that you can see already just by looking at this figure, that there are some populations with more lines and more stars. So these reflect 6 populations that have evolved approximately 100-fold higher point mutation rates in the LTEE. And this observation allows us to set up something very beautiful, a beautiful hypothesis test, okay? We would expect hypermutators to accumulate more mutations in anaerobic specific genes, reflecting relaxed selection. Again, so this is just the idea that these genes aren't being expressed. You can't purge deleterious mutations. So essentially they're like buckets for mutations. And in contrast, for our non-mutators, we would expect mutations to accumulate in aerobic specific genes, using that loosely, reflecting positive selection, okay? And I will say conventional wisdom suggests that we are not able to tease apart selection using the hypermutators, but I'm going to show you here in this work that that is not true. And if you're interested in how we were able to apply that and figure that out with other decompositions within the LTEE, as Rich said, there's a paper on bioRxiv that was posted yesterday. So here, what I'm just showing you, I'm showing you the number of observed mutations at one time point at 50,000 generations in our aerobic gene sets and in our anaerobic gene sets. And what you can see already here, there's about 3 times more mutations in the anaerobic gene set as expected, reflective of the relaxed selection. And in contrast, when we look at our non-mutators, so these are strains that have retained the ancestral point mutation rate, we see that there are more mutations in the aerobic gene set than in the anaerobic gene set. These are significant differences, and this then reflects positive selection, okay? So this clear contrast between the hypermutator and non-mutator population suggested that one can get better resolution of the selection dynamics in these sets, given an individual were able to compare the observed mutations to some expected number of mutations within each population. And it was with that thinking that a simple test to infer mode of selection, what we call STEMS, was born. And the idea behind STEMS is relatively straightforward. The thought is that we can randomly sample N mutations within the genome where N has the same cardinality of some gene set of interest. So in our case, the aerobic or anaerobic specific genes. And then we can integrate those mutations over time to generate a node distribution of the expected number of mutations within that population. Now, when we compare the mutations that we observe to this node distribution, as you can imagine, you can come up with some different outcomes. Now, if the mutations that we observe in each one of those populations fall within this node distribution, that is what we call obscured selection. You can't tell what is going on in that instance. However, if the observed mutations fall below that node distribution, that would be evidence of purifying selection. And of course, as you can imagine now, if you fall above that node distribution, then that is a signature of positive selection, okay? So on the next slide, I'm actually going to show you some data in which I was able to apply this to the genetic decompositions, the aerobic and anaerobic gene sets in each of our populations. I'm gonna show you the aerobic specific genes in black, and I'm going to show you the anaerobic specific genes in red. So this is in line with the same colors that I used on the last couple slides ago. And here in the top 6 panels, I'm showing you the non-mutators. And what you can see from this figure is that in all instances, the black curve falls above this node distribution. And that means that this gene set is under positive selection. And these are our aerobic specific genes. And just to kind of orient you a little bit more, I'm showing you the cumulative number of mutations normalized by gene length on the y-axis. And I'm showing you the generation number here on the x-axis, okay? Now, in our anaerobic specific gene sets, again, we expected more mutations to accumulate in those gene sets. And this is exactly what we see. We actually see a couple of things here. I'll put it that way. So we see that both the aerobic specific gene sets and anaerobic specific gene sets fall within this node distribution, right? So this would, considering what I showed you earlier in the model, that would say that selection is obscure. However, more often than not, we see that there are more mutations in the red because it always falls above the black line, except in a couple of populations. Now, there's a simple explanation for why we see the aerobic specific genes accumulate mutations at the same rate, and that is called emergent neutrality, whereby mutations, they accumulate on backgrounds, on beneficial backgrounds ever so slightly. So essentially, you're bringing these essentially deleterious mutations up with you into this regime. We also see that in a couple of populations, AR-1 and AR-3, for example, the black curve falls above the node distribution early on. Now, this just reflects the fact that these two populations evolve hypermutability later on. So we can see in these populations that we are able to see evidence of positive selection, as we should when they have the ancestral mutation rate. And then later on, we see them with the relaxed selection, depending on their regime. Now, interestingly, with this method, we were also able to resolve relaxed selection. Purifying selection. So here in the aerobic specific gene sets, we see that they fall below this non-distribution. So STEMS, unlike conventional wisdom would suggest, is able to resolve relaxed, purifying, and positive selection in both the non-mutators and hypermutators as well. So after looking at the global response, the global mutational response, I want to center my attention a little bit more on genes known to regulate the aerobic to anaerobic metabolism switch. And it is known that 3 proteins are responsible for that: fumarate and nitrate reductase protein, which responds to oxygen directly, and ARC-A and ARC-B, which make up the two-component regulatory system that responds to the redox cycle of the cell, redox state of the cell. And this mechanism is quite beautiful, so I'll just run through it real quick. So ARCB is the sensor kinase, and it undergoes what is called autophosphorylation, which just means that it can pick up a phosphoryl group from ATP and then add it to itself. That phosphoryl group is then sequentially passed down through multiple domains within the protein, then terminating on ARC-A, which is the response regulator that can then go on and activate or repress genes within its regulon. On the same coin, ARC-B also has phosphatase activity, which means that it can remove this phosphoryl group, sequentially pass it back upwards in the protein, and then dump that as inorganic phosphate within the cytoplasm. Now, when we look at mutations in FNR, ARC-A and ARC-B, there's nothing really going on in FNR. One population has a mutation that pops up at least at 30,000 generations. And I look at these at 10,000 generation intervals, and I didn't see anything after that in that single population. However, when we look at ARC-A and ARC-B, we see that all populations have a mutation in either ARC-A or ARC-B, and that's shown here in yellow. Now, there is one instance that I have twice. So I have error -2S, which just means small, and error -2L, which just means large. And this reflects two ecotypes that have been coexisting in this population, one an acetate specialist and one a glucose specialist. So how we interpret this is that the acetate specialist has evolved a mutation in ARC-A, whereas the glucose specialist has not. Now, these mutations, you can ask whether or not they're non-synonymous, whether or not they're important at all to this function at all, to the function of the protein. Now, I'll say that they fall within protein domains that are relevant to either the transfer of the phosphoryl group, so I'm showing that here in ARC-B, or also the sensing of the redox state of the cell. So you can ask then, are these mutations breaking the ability of E. coli to respond to aerobic or anaerobic growth conditions, right? What are the effects of these mutations? Now, to answer this question, it is known that ARC-A and ARC-B mutants, that are deficient in signaling, are sensitive to photosynthesizing dyes. And these are dyes that just increase the concentrations of ROS within cells. So what I was able to do, I was able to make some media, add some of that dye to the media, and ask what is the phenotypic, what is the growth response of the evolved populations on that plate. So here I'm just showing you an ARC-A knockout, the ancestor strain REO606, and an ARC-B knockout, and these are single knockouts. And I'm showing you each one of those populations here on this axis, and I'm showing you the generations, 2,000, 10,000, and 50,000 generations. Now at 2,000 generations, none of the populations had mutations in ARC-A or ARC-B, and we can see that these strains grow confluently across their lawn, which means that they're not sensitive. And just to kind of go back to say what a sensitive strain looks like, it forms these punctuate dots or don't grow at all. At 10,000 generations, 75% of strains with ARC-A mutations are sensitive to this treatment, and 67% of ARC-B mutants are sensitive to this treatment. At 50,000 generations, 86% of ARC-A mutants are sensitive to this dye treatment, and 100% of ARC-B mutants are sensitive, again, as shown in some instances where there's no growth. Now, if you know anything of our system, you know that these populations have evolved in this minimal glucose media, and thus growing on other media they don't do so well. So one can just remove the dye and ask whether or not the effects we are seeing is a plate effect or a dye effect. And that's exactly what I did. And we can see that at each time point, I was able to rescue that sensitivity, except at 2 time points, error -2 at 50,000 generations and error +6, though error -2 does seem to perform better, okay? So far I've shown you that ARC-A-B signaling has been altered, which implies that the evolved populations are highly susceptible to oxidative stress. But I'm in the Lenski lab, and I have that whole freezer. And one cannot be in the Lenski lab without doing a fitness competition at all. So that's what I did. I did fitness assay in an oxic environment and then in an anoxic environment, okay? And to make my media anoxic, I just made it anaerobic by flushing through some gases, and these competitions were done in an anaerobic chamber. So we would expect, because the populations evolved in the oxic environment, that they would be more fit in that environment. And that is exactly what we see. So I'm showing you oxic fitness here on the x-axis, and I'm showing you the fitness in the anoxic environment on the y-axis. And as you can see, each one of the points fall below this isocline, demarcating the two environments. So thus they're better adapted to this environment. And what the colors just show, it shows that the populations, when they became mutators, so that you can see what the effects of hypermutability has on fitness. Now I can then summarize this just a little bit more, and I can look at the grand means of all of these populations in both of the environments to see the temporal trajectories of fitness. And we would expect, again, fitness to increase in the oxic environment. They evolved in that, and I've shown that here already, but we'll just go on and look at the grand mean trends. And we can see exactly that. We see that in the oxic environment shown in black, fitness at 50,000 generations, at least in my studies, are about 50% higher than the ancestor. And here in the anaerobic environment, we see that fitness increases through 10,000 generations, significantly so in the first 10,000 generations. And interestingly, even at 50,000 generations, these strains have not lost their competitive ability against the ancestor. And again, this is extremely surprising, especially given the fact that we have mutations. We see evidence of relaxed selection in those genes. So they're accumulating mutations. And as we know, mutations are mostly deleterious. And also we see mutations that affect RB signaling. Again, I just showed you that in the previous slide. So this to me suggests that anaerobic metabolism is buffered against the effects of those deleterious mutations, right? And it kind of makes sense when we think about how metabolism evolved. Life first evolved in an anoxic environment, and organisms living in that environment did so for 2.4 billion years before aerobic metabolism evolved. So given the fact that I saw this correlation in all of these gene sets, I thought to myself, well, maybe the gene sets that we identified as being strictly aerobic and anaerobic gene sets, and again, this was identified by another group, right? Are they connected in any type of way? So that was the question. I just asked, is there evidence that aerobic and anaerobic specific genes are connected, right? And to answer that question, I used a database called String, which is just a database of protein association networks that uses 7 lines of evidence to predict interactions between genes, okay? And these 7 lines of evidence can range anywhere from empirically derived to computational derived predictions. And this is what we see. So here I'm showing you a network with the aerobic and anaerobic specific gene sets. And as you can see, they are very well integrated. They're not single at all. So there are other genes that was off of the network, so not integrated in this network. So of course, there are some genes that exist by themselves and only connected to, their specific partners in a gene set. But again, this just shows that they're more connected. Now, the fact that we saw the mutations in ARC-A and ARC-B, evidence of relaxed selection, and this increase in fitness means that something is going on, some type of correlation. And I think the data I've provided so far in this chapter lends evidence for something called buttressing pleiotropy that has been described as such. So it's defined as the function of an unselected trait is being maintained by selection on the trait that it is correlated with. Now, there isn't much evidence of this that has been described, let's say, biologically, but I think the evidence that I provided actually kind of holds in this sense. All right, so that's chapter 1, the end of chapter 1 material. So I'm going to close the door on that. And then I'll open the door on my chapter 2 material on cell morphology. All right. So cells come in different shapes and sizes, but why they adopt these characteristic morphologies are widely unknown. Why, for example, did E. coli evolve a rod shape, whereas Corynebacterium evolved like it looks like it evolved to play a doubles match with Venus and Serena Williams? Of course, these characters generally reflect specific adaptations to the conditions that these organisms evolved under, but nonetheless, the transitionary morphs made during those periods are unknown. Now, when we look at cell size, we see that larger cells are associated with greater biological complexity. Now, one argument for why this occurs is because larger cells can house more DNA and thus more instructions. But this does not come without a cost. So we've all heard this argument before about surface to volume ratio limiting cell size. And this because the volume of an object generally increases faster than its surface area. Now for eukaryotes, they can combat that owing to the fact that they have complex vasculature systems and transport systems that can get things from point A to point B and back and forth. However, for bacteria and prokaryotes, this is problematic because they rely solely on diffusion, which makes it hard to import and export nutrients and waste products respectively. Thus, in terms of cell size, the effective evolutionary strategy is to maximize surface area to volume ratio by decreasing cell size, okay? And that is why it came as a surprise in the first 10,000 generations when we saw that fitness, here shown in this first panel, and cell size, they increased simultaneously and together, and they were very well correlated, okay? Now to fast forward 40,000 additional generations from this point, we know that fitness increases by a power law, which just means that fitness will increase indefinitely. So this is just work done by Weiser et al. And what I'm showing you is that the fitness measurements, they line up with this blue line and not this red line here, which has an asymptote. So this means that fitness will increase indefinitely, right? So given this observation and given the earlier observation that cell size and fitness had increased, the motivating question for chapter 2 is, do cell size and fitness still correlate? Now to answer this question, I went back to the freezer. I sampled population at 2,000, 10,000, and 50,000 generations, the clones and whole population samples, grew them up for a couple days, and then I measured cell volume by 2 methods using a culture counter, which measures the volume of a cell as it breaks the current through an aperture, a small hole. And I also measured cell volume using microscopy and cell shape as well. Now, for the microscopy, one can use machine learning, and that is what I did to process those images. And what machine learning allows you to do, it allows you to segment each individual cell on a micrograph, and then it gives you a measurement of length and width, and also volume for your cell. Now, when we look at the concordance between microscopy and the culture counter, they're highly concordant, right? So I'm showing you they have a strong association here. Culture counter volume on the x-axis, and on the y-axis, the volume integrated by the microscopy, and we can see they're very strongly correlated. So when I show you data in the future, when I'm referring to cell volume, I'm referring to volume measured by the culture counter because I don't need to show you both. And when I'm talking about shape, these measurements were estimated using the microscopy and machine learning. So here I'm just showing you that cell size increases in idiosyncratically among clones. So these are clones. I'm showing you cell volume. I'm showing you each one of the populations, and again, at 3 of the time points. And what do I mean by idiosyncratically? Instead of increasing without pause, we see that in some populations, chiefly error -2, error -3, and error -6, we see that the cells decrease in size between 2 and 50,000 generations, so at the 10,000 generation points. But we know that clones they're not always representative of the whole populations they're sampled from. And if that is the case, we would expect to see a more consistent temporal trend when we look at cell size in whole population samples. And that is exactly what we see. We see that cell size increases monotonically in these whole populations. So without pause, we see that in each of the populations, those cell size increases. And when we look at the grand mean and 95% confidence intervals for each one of these time points for the populations, we see that that occurs and is significantly so. All right. Now, I'm also a microbiologist, and these assays were done with cells in exponential phase. So one would ask, does the same cell size trends hold for exponentially growing cells? Okay. And we'd expect a couple of expectations. Cells are usually bigger during exponential phase for a couple of reasons. One, because bacteria, they have multiple copies of their chromosome, so they're getting bigger just to accommodate those chromosomes. And secondly, they have more ribosomes within the cell to accommodate the faster growth rates. And that's exactly what we see. So here I'm showing you the median cell volume at 50,000 generations for each one of the populations. And we can see that exponentially growing cells in black are bigger than the stationary phase cells that we measured. Now, when we ask how well they correlate with each other, so how well stationary cell volume predicts exponential cell volume, we see that they're strongly correlated as well. And we see that there's approximately a twofold difference between exponential and stationary cell volumes, implying that at least the cells undergo at least one reductive division event. Excuse me. So I've shown you so far a lot of data on this chapter on cell volume, but what about cell shape? So let's just talk about that real quick. So for a cell, assuming constant volume, increasing cell length is more beneficial. This is because, again, that surface area to volume ratio, if you get 2 times bigger, you have a 50% lower surface area to volume ratio, and the lower is the worst. And if you get longer, you have a 4% lower surface area to volume ratio. Now, if we hold cell shape constant, increase in volume decreases the surface area to volume. And this is what we have been seeing. We've seen an increase in cell volume. So one can then put forth a hypothesis that seeing that the cell volume has gotten bigger in the long-term lines, that the cells may have or should have gotten longer to compensate for a reduced surface area to volume ratio. And we actually see the opposite. So here I'm just showing you the 2 ancestors, REO 606 and REO 607 at the bottom. And as you go up this tree, we're increasing in the number of generations. And what we can see is that all of the points, they most likely fall within the first 10,000 generations to the left. This means that the width is getting bigger, so they're getting wider. And then by 10,000 generations, they shift more over to the ancestral aspect ratio or further on to the right. Now, again, when we look at the grand means and associated 95% confidence intervals, we see exactly the same trend. Within the first 10,000 generations, they get wider. And at 50,000 generations, they get longer. And there's no significant difference between the time at 50,000 generation and the ancestor strains. So there's this evolutionary reversal of aspect ratio. Now, one can ask then, given the surface area to volume requirements, whether or not this reversion is to the benefit of increasing that surface area to volume ratio. So you would need to calculate that quantity. And this is what we see here. So this figure is in the same style. And we can see that the mean surface area to volume decreases through 50,000 generations. And in all cases, it does significantly so. So no, they have not increased their surface area to volume ratio at all, it appears, with increase in length. But one can test that formally with an isometric analysis. Basically, what one can do, you can compare the surface area to volume ratio at 50,000 generations. So if surface area to volume ratio, presuming aspect ratio had not changed between 10 and 50,000 generations. And what we can see is that with no shape change, you have a surface area to volume ratio of about 0.33. And with the shape change, it does increase, but not significantly so, all right? Now, on shape again, in one population, it evolved what is called, what we term stubby cells. So here, this is era +5, and E. coli typically maintains a rod-shaped morphology. And you can see that in some instances, they're clearly spherical, right? So this is non-traditional E. coli cells. And those are shown here in 50,000 generations. And the fact that they changed from these cockeyed-looking cells or these stubby cells to the natural E. coli morphology suggested that there was some genetic determinant for this trait. Okay, so to look at that, I looked at evidence for mutations in morphogenic genes. So these are genes that are known to be associated with changes in cell shape, and this is what I'm showing you here. So I'm showing you at 2,000, 10,000, and 50,000 generations, the type of mutations that occur in these populations. And we can see that all but one population, era -5, have evolved mutations in these morphogenic genes. Now, these mutations are all non-synonymous mutations, except in populations that evolve hypermutability. And we also see that the stubby era +5 population evolved a single mutation in MREB. It maintained that mutation all the way through 50,000 generations. So if you know anything about cell morphology, you know that MREB, cells depleted in MREB, they become spherical. And the fact that we only see this mutation in MREB makes it a high candidate that this mutation or this gene is causing the phenotype that we are observing. And it also suggests that at 50,000 generations, there's some other gene that is compensating for the change in cell shape that we haven't identified here, at least within these 5 genes. All right. So now to the main question that I started with in this chapter: do fitness and cell size still correlate? I bet that you already know the answer, given the fact that we've seen cell size have increased, and the answer is yes. Fitness and cell size still correlate, and they're significantly so with some scatter. Now, I couldn't show you all of this data without showing you any pictures of the populations. So here are these are clones from each one of the populations. Here are the ancestors, REO 606 and 607. And here are clones from the populations at 50,000 generations. And you can see that their serum morphologies are extremely heterogeneous. And what you can see is that these clones are, they're massive. They're bullies compared to REO 606. But also what you can see, if you're me, you also see that there's an anomaly in one of these populations, era -3. You see that instead of having these clear, nice, big black cells, they also have these indications in the background, which I called ghost cells, okay? And it was with this observation that I opened the curtain on my chapter 3 talk on mortality. All right. So the media that the clones or these populations have been evolving in for the last 70,000 generations has abundant glucose, and it also has citrate that is metabolically inaccessible to the ancestor. Now, E. coli doesn't encode a transporter for citrate under oxic conditions. However, in one single population, there was a duplication event of a transporter. That transporter was duplicated and put in front of a promoter that is responsive to oxygen. So now this transporter can be expressed in an environment with oxygen, okay? And because of this mutation, there is a approximately six-fold increase in population size. And I'm just showing you that here by the turbidity differences between this population and the other ones next to it, right? So this became our CYT+ strain. Now, given the fact that we saw these ghost cells in that population, and this is the only population that evolved this unique metabolism, it makes sense to say, well, maybe this morphology is associated with the citrate use. So I asked, are these ghosts of citrates passed? So here I'm just showing you the LTE ancestor again, REL 606, grown in DM25, and I'm showing you that CYT+ clone grown in the same media. Now, because this clone can utilize citrate, you can remove the glucose from the media, force it to grow on citrate, and that's what I did in preliminary experiments. And lo and behold, I was able to exaggerate that phenotype of those ghost cells, right? So this suggested that growth on citrate was at least associated with this cell morphology, all right? But I didn't know whether or not these cells were dead or alive, and I wanted to know that. So what I used a simple assay, which allowed me to stain cells that were alive green and stain cells that were dead red, right? And then I can quantify that again using the machine learning software. And that's what I did. So here I'm just showing you that CYT+ clone, and I'm showing you the ancestor, and I'm showing you a raw image here. I'm showing you in this image cells that were stained green, so alive, and I'm showing you cells that were stained dead here in red. And then I'm showing you the composite of all 3 of these figures here to the right. And what you can see clearly that there are a lot more red cells, a lot more dead cells in REO 11364, our CYT+ stain. There is some in the ancestor, but we can also quantify this, and that's what I did. So here I'm just showing you the proportion of cells, and in REO 606, we see about 13.2% of cells die in that environment, whereas 43.6% of cells in the of CYT+ cells die in that same media. So this suggests then that those ghost cells that we're seeing are cells that are actually dead. So when does this elevated cell mortality evolve? So to answer that question, I examined clones spanning the entire era -3 phylogeny before and after CYT+ evolved, right? So at each one of these time points, and this is what we found. So here I'm just showing you the proportion of dead cells on the y-axis, and on the x-axis, I'm showing you essentially a CYT+, era -3 clone during its evolution between 0 and 50,000 generations. So this is just time, okay? And what you're going to see is a couple things. So CIP plus evolved here at this point, 31,000 generations, and it was initially weak. And just to show you the mechanism of that, so it evolved this CIT transporter under the control of that oxic promoter. And citrate works by, well, citrate enters the cell by exchanging succinate, fumarate, and malate out of the cell, okay? And as we can see here at 33,000 generations, we see a high elevated cell mortality. And this point is actually interesting because it was at this point the citrate trait was refined. It was at this point it got better at using the citrate, and this is why. It evolved a mutation in the regulatory region of this protein called DCTA, which takes succinate, fumarate, and malate and brings it back into the cell. So now you get the cycle of CIT pumping succinate, fumarate, and malate out to bring citrate in. And then at the same time, DCTA bringing those equivalents back in. So you get this cycle of growth to consume all of the citrate, okay? So having seen that, one can ask then now, does genetic background play a role in this elevated cell mortality? So Zach Blount, he is the king of this citrate guy. So any kind of strain that you need, he has them. He just has them somewhere, right? So here are CIP plus clones, and he isolated some CIP minus revertants. So these are clones that have the same background as the CIP plus clone, but they cannot use the citrate. So you can just use this to test the effects of the background itself, other mutations that are occurring in the background on the cell mortality. And the thought is that if growth on citrate alone correlates with elevated cell mortality, then we should see less death in the CIP minus revertants. And that is exactly what we see. So here I'm just showing you CIP positive cells in red and CIP revertants cells in blue. And you can see that more oftentimes than not, we see that cell mortality decreases in the clones once the ability to utilize citrate is gone out of the cell. So now I have some conclusions just to go through. And I think it's kind of difficult to do that with 3 typically very, very unique chapters. So I think what sums up all of this work, and I don't have any words on the slides, and you'll find that out soon. What sums up all of this work is that 3 words. It's like, I don't know. I found evidence of correlations in aerobic and anaerobic metabolism, and I see that fitness has increased. We see that cell sizes continue to increase and that it tracks well with fitness. And we also see that with using citrate, there's also elevated cell mortality. Now, Charles Darwin himself said that these correlations were mysterious. Now, my time here at MSU is done, and I would have liked to find out why am I seeing these correlations and I cannot. Nonetheless, I think the data I have shown you today highlights the fact that these correlations do exist, and there's still work that needs to be done. And with that being said, I would like to just acknowledge my dissertation committee, my undergraduate mentees, Joe Warren and Ali Abdel Majid, members of the Lenski Lab, and my collaborators. And I don't care how good a student is, one must always acknowledge the support staff in their department who make sure that things just work well. Now, I told you I had 3 chapters on M, but I have a little bit of time left. So we have a bonus chapter on M, and this is going to be on mushy stuff, right? Because we need some mushy stuff at the end as well. And I'm gonna start here with this picture. A picture of my grandmother and my grandfather. And I'm just going to talk about opportunity. My grandmother, she immigrated here from Guyana, South America in 1973. And then she brought her husband along, who then made way for their 10 children to come, who then made way for over 20, 30 cousins, who then made way for so many grandchildren. I have a very, very loving family. They've supported me. They've cared for me. They've called for me. There are times where I was in trouble and I always had a place to go. And they've just always been a support system. We take people into our family, no matter like in-laws, friends, whoever, as though they were always part of us. I would also like to thank these two people. My mother, who never gave up on me. And I said I wasn't gonna cry, but I was a horrible kid growing up, and she never gave up on me. She made sure that I hit the books. She made sure if she had to lock me up, she locked me up. She made sure if she had to lock the doors, she locked the doors and said, hey, you have to respect my household. She held me responsible for the things that I needed to do. And also my stepfather, who came into our life, I call this man my dad. He came into our life in 1995. My mom had 4 children, and he took us in as though we were his own. And he never left, even though I almost killed him one day by dropping a car on him when he was working underneath it. So that's kind of funny. I also have 5 amazing siblings, all doing something with themselves, loving, caring. We've had our days, but we've loved each other. We're all doing our thing, and I think it's just amazing that I'm here and I'm able to represent all of them as I go on this journey. I would also like to thank my beautiful wife, who stepped into my life and is helping me to raise 3 beautiful children. She's my friend, she's my partner. She makes sure I eat well. She's taken out more trash in the last 2 months to make sure that everything was fine so I can study and prepare for this defense. And I love her greatly. That's my best friend. I also shared 2 children with this woman, Dominica. I would like to thank her for always being there for the children and making sure that they have what they need. Make sure that when I'm stressed, that I can send them that way and she take care of them and do what they need to do. I put 2 feet down. I would also like to thank my children who have been on this journey since my time at Grand Valley. They have kept me focused. They've made sure that I don't drink too much coffee because it gives me headaches and nosebleeds. They have listened to me say 10 more minutes, 10 more minutes, 10 more minutes so many times until the fact that they fall asleep on the couch waiting for me. Kids, this is for you. I love you all, and I hope that me being here today is proof that people can make it despite where it is that they come from. And to let you all know that you will be challenged in your life because challenges come with life, just living. And I want you to know that despite those challenges, to keep pushing, just like your dad did. I would also like to thank Rich, who is an amazing individual. He's very knowledgeable, he's very smart, but he's also very humble and caring. He cares more about the person. He cares about your research, but he cares more about you. I've been through significant challenges here in graduate school as well. And there's times where I just came in, not myself, just dragging. And Rich said, go home. And I don't know if any advisor would have done that. I wouldn't have made it here without his willingness to say, hey, take care of you. I would also like to thank the people that I've met in the lab, having fun, drinks at Crunchy. When I was drinking, Johnny Vegas was my best friend, but we ain't gonna talk about that. The people that I've went out to play games with, went to the gym with, just hung out with, and just kept it cool. I appreciate y'all. I appreciate the fun times, and I hope that beyond graduate school that we can remain friends. There's also another thing I would like to highlight, and that's the opportunity that my kids have got to have since they've been here. My nieces, nephews, anybody I've exposed them to the lab. You know, I'm an African American, I'm Black. I'm gonna say I am Black. My family's from Guyana. And oftentimes you don't see Black scientists, you understand? So at any given point, I was able to bring my children to school, and they were able to see what science is. And because of that, my daughter wants to be a scientist. My son is thinking about being a scientist. And that is the type of change opportunity brings to people. And here's my Lenski family. These are all of Rich's trainees. I am glad to be here in the back. I'm glad to represent the lab. And with that, I can take any questions.
All right. Nkrumah, that was a brilliant talk, and I'm letting people gather their questions. I think we're all incredibly moved. I'm seeing people, fantastic talk in the chat session. What I'm going to do, though, before we go to the, everybody be thinking about your questions, but I'm going to try to click on this button that says unmute all. And when I unmute all, let's have a round of applause for Nkrumah for a fabulous presentation.
Great job. Great job.
Thank you.
Well done.
Fantastic. So let's see if I can do it all without turning Nkrumah off. And Jay, maybe you can help me. Let's see. So if there's a raise hand kind of option, and let's see how this works. We'll see what we can do. And I'm seeing amazing job, great job, awesome, awesomely beautiful. It's incredible. So let's see. I see a question from Joe Graves. So I'm going to unmute him if I can figure out how to do that. Joe, you should be unmuted.
About your CIT plus lines, your supposed ghost cells, whether in fact what you're looking at is a trade-off between their ability high classic laboratory trade. Did you hear my
We're
question?
not hearing you.
I'm sorry, Joe. You kind of cut out in the middle of your talk. So I mean, in the middle of your question. So I didn't quite hear you. I did hear that you say something about a trade-off with CIT plus and something. I didn't know what else you were saying.
Yeah. So yeah, Ed, Rich.
Oh, you try again, and if you can't hear this time,
Okay,
then
so
I'll
what
try.
I was wondering is if because they're growing so growing rapidly on CIT plus, that in fact since their lifespan is shorter, and so you see a higher mortality rate. So it's a sort of classic trade-off between rapid growth and lifespan.
Yeah, so I mean, I think that's definitely an option. The fact that, I mean, we've kind of thought about this in terms of saying maybe the cells are growing so fast on the citrate, they kind of hit a wall. They can't really switch their metabolism between glucose and citrate metabolism. In terms of kind of this trade-off, I think that's a great hypothesis, a great idea, and it's something definitely that could be addressed in future studies. Again, so I don't really know. So this is the last chapter. This came like literally within the last year, and I had some other things going on. So there's a significant amount of work that needs to be done in that system.
All right, let's see. I see Jonathan Hardy has a question, so I'm trying to unmute him. Go ahead, Jonathan.
Can you hear me,
Yes.
Nkrumah?
Yes, I can.
Yeah, I was wondering, the ghost cells look very interesting to me, and a lot of these live cell, dead cell stains are based on permeability. And so they look like they might be more permeable. And so I'm wondering if you believe that they actually can't replicate, that they can't grow.
I'm gonna say,
is there any way to test that?
Yeah, so there
That's
is
separate from the permeability. Do you know what I mean?
Yeah, so so you are correct to say that the live dead assay is a permeability assay, right? So even when we look at the ancestor, we see the cell death. When I look at where the dye is staining in those cells, they look to be staining as almost always associated with cells that are about ready to divide, right? So that suggests that's where they're at the synapse, for example, where they're going to divide, that's where the stain is going. And that may be reflective of the permeability. Now, one can actually measure cell death directly by measuring lactate dehydrogenase. So this is something that is found in the cell itself, in the cytoplasm, and only released when the cell dies. So that's definitely something that I would like to see done in the future to test that directly.
Beautiful job.
Thank you.
Congratulations.
Right, former lab mate Alita has a question, and I've just unmuted her.
And mortality, it looked like the patterns that you observed varied depending on if you assayed with clones versus whole populations. And I'm just wondering if some of those patterns might be driven by the ecology or frequency dependence, and could you see different distributions in populations that have frequency dependence? And I guess the big question is, might frequency dependence lead to the morphological changes even in these microbial populations?
I think it could definitely lead to those changes, like the ecology itself. I haven't tested that with whole populations yet, but I have good thoughts that I think the cell mortality may go down in the context of the whole population samples. That's just my thought, though. I haven't tested that yet, but I have a good feeling about that.
Yeah, that would be my hunch too. Thanks.
Yeah.
All right. A question from John Williams, so I'm going to unmute him now.
Great talk. So when you were talking about the fitness of aerobic versus anaerobic growth conditions, are you talking about anaerobic respiration?
Good question.
Versus fermentation?
So that's a very good question. So actually, so when I grow these strains in an anaerobic environment, we grow them in the same media that had just been made anaerobic. So we don't provide them with an alternative electron acceptor. So these strains are presumably doing fermentation. And that to me is kind of interesting because in the long-term environment itself, just like by virtue of just growing in the long-term evolutionary experiment, the cells, we know that they have increased their growth rates, for example, and some of them have evolved cross-feeding on acetate, which is a product of what is called overflow metabolism. And that overflow metabolism is resultant of fermentative growth, right? So I think that actually serves as a good hypothesis for why we see growth in the anaerobic environment increase, is the fact that they're doing fermentation in both environments. So that's a good question. But it's definitely something of interest to test growth using some of these alternative electron acceptors.
Okay, thank you.
All right. I see future lab mate, Eric Bruger. I'm unmuting him now.
Hey, Nkrumah, nice talk.
Thanks, Eric.
I also had a question about the cell size data.
Okay.
And I was curious if you have any idea if there is much plasticity in that trait, particularly as possibly a function of substrate availability. And I was wondering if you had done any tests like under different carbon amounts or conditions to assess that.
You know, I have not. So I just recently came across some work that showed that depending on the substrate that you're using, if it's a high quality substrate, that you get this kind of plasticity, like different cell sizes. I have not done those experiments. So I haven't done, I would say, tested any type of hypothesis specifically with cell size. I think Rich would tell you that, I mean, that project was extremely, it's expansive. I mean, it's a lot of work. But that's definitely something that could be tested in the future. And I think that may be associated. I think so cells, they're getting better perhaps at using the glucose. They are getting better at using it. And perhaps that can be equivalent to kind of saying that their substrate is better. So I don't know. It is a good thought though.
All right, cool, thank you.
Yeah.
All right, I'll give a second. I don't see any more hands. And Nkrumah, you had 126 people here. I'm going to unmute everybody now, just to let everybody know that the day's procedure is that poor Nkrumah, he's going to get a few minutes rest after this, and then his committee is going to meet with him, and we'll have a chance to ask him some more questions. But as you all know, it was an absolutely fantastic talk. I'm getting ready to unmute everybody, and let's give Nkrumah another hearty round of applause and shout out your congratulations.
Oh,
Yeah.
Thanks.
Mr. Grant, I'm gonna get my couple last
Bring
Mr.
up
Grant in there.
Nkrumah's picture again. Where's Nkrumah here? Nkrumah, so we can see you.
Good job, Nkrumah.
Thank you.
Wonderful presentation. Wonderful presentation.
Wasn't it great?
Thank
Wasn't it amazing?
Thank you.
Yes,
Thank you.
very informative, and easy to understand. So I appreciate that.
Thank
He's
you.
a brilliant scientist.
Absolutely.
Brilliant.
Thank you, everyone.
I look forward to seeing the
Hi,
next.
Miss Russell. Hi, Amy. Thank you so much for coming.
So proud of you.
I thought this presentation was going to be all suit, but the science was pretty good.
Great work, Nkrumah.
Thank you. Thank you.
Madeline's clapping too.
Daddy did a great job.
Awesome. That was so good. I'm freaking so much.